Get ready for a genetics adventure. From Linnaeus's classification system to Mendel's pea plant experiments and the DNA double helix, we'll explore the incredible breakthroughs that unlocked the secrets of heredity. It's a story of brilliant minds, groundbreaking discoveries, and the amazing journey to understanding the very blueprint of life.
Influenced by his father's botanical interest, Linnaeus cultivated a love for plants and from an early age. He created his own garden by the age of 5. Linnaeus authored numerous works, with his time monitory being one of the most significant, laying out a comprehensive framework for classifying life forms. He introduced the binomial nomenclature system, making it easier to identify and classify species, thus resolving confusion in early classification methods. His trip to Lapland resulted in a documentation for over 100 new plant species, forming the foundation of Flora Nymphalica. In 1761, he was knighted by King Adolf Frederick, adopting the name Carl von Linnaeus, reflecting his achievements in the scientific community. The Linnaeus Society, founded in his honor, continues to promote natural history, ensuring Linnaeus' contribution to science are preserved. Carl Linnaeus' life and works encapsulated a transformative period in the history of science. His legacy is a testament that enduring the value of curiosity, exploration, and systematic inquiry in understanding the natural world.
Charles Bonnet was a Genevan naturalist and philosopher in 1720 and 1793. He proposed the idea of preformation, suggesting that organisms develop in the miniature versions of themselves. So I have mentioned about the idea of Charles Bonnet, which is the preformation. Um, Preformation theory states that the miniature human called homunculus has already preformed in the egg and the sperm. Homunculus contains from the sperm cells of the male and the egg cells of the females. And according to Charles Bonnet, each sex cell contains a complete miniature versions of the adult organism, which someplace and grows and develops during gestation. Charles Bonnet, he believed and he advocated about the preformation theory because living organism has already preformed within the egg and the sperm.
Good afternoon, everyone. Good afternoon, ma'am. I am Rika Jinti Saha. I am the third person to discuss the history of Gregor Johann Mendel. Gregor Johann Mendel was born on July 20, 1822, and also he died on January 6, 1884. Mendel was an Austrian meteorologist in 19th century. He is known as the father of modern genetics for his groundbreaking work on heredity. Mendel conducted experiments of pea plants in the garden of his monastery, where he discovered the basic principles of inheritance. He showed that the traits are passed down from parents to offspring through discrete units, which we call genes. Mendel's work laid the foundation for our understanding of genetics and evolution. That's all, and thank you.
Franz Ulmer contribution related to Mendel studies. Franz Ulmer is a 19th century botanist and paleontologist. He proposed that plants and animals are evolved over time due to environmental influences, but his theory lacked a clear mechanism for inheritance. Gregor Mendel worked on pea plants, provided the missing link by demonstrating that traits are inherited through discrete units, known as the genes. rather than blending together. Mendel's principles of dominant and recessive traits, segregation, and independent assortment explain how genetics variation could persist across generations. Mendel's finding thus completed Unger's evolutionary framework by providing a scientific basis for heredity and laying the groundwork for later evolutionary theories, including the modern synthesis of Darwinian natural selection and genetics.
Hugo de Vries was born in Haarlem, Netherlands. He was a professor of botany at the University of Amsterdam when he began his genetic experiments with plants in 1880. Hugo de Vries made a big impact in helping the world recognize Gregor Mendel's work on genetics. In the 90s, something nagwajo sa kagaling eksperimento sa plant hybrids. De Vries discovered patterns in inheritance that were very similar to Mendel's findings from decades earlier. Around the same time, two other scientists, Carl Correns and Erich von Tschermak, made similar discoveries. When De Vries first published his results, he did not mention Mendel. However, after learning about Mendel's earlier experiments, he acknowledged Mendel's work in his later publication, written in German. This helped bring Mendel's forgotten research back into the spotlight. De Vries didn't just rediscover Mendel's work, he also added to it. He proposed the mutation theory, which suggested that new traits could appear in plants and animals through sudden and large changes or mutation. This idea explained how species could evolve over time, complementing Mendel's law of heredity, which focused on how traits are passed down from one generation to the next. This work, combined with Mendel's, helped establish the field of modern genetics and deepen our understanding of how traits and evolution work.
Carl Erich Correns, 1864 to 1933. Carl Erich Correns is a German botanist and geneticist. He began his research on trait inheritance in 1892. He was initially unaware about Mendel's law of heredity, but with his own experiments with bee plants led him to the same conclusion as Mendel's. In 1900s, Correns published his findings in a paper titled G. Mendel's Law Concerning the Behavior of the Progeny of Racial Hybrids. Correns didn't just repeat Mendel's work, he also added his own brilliant discoveries. He conducted a research on cytoplasmic inheritance, an important extension of Mendel's theories. He studied the plants with unusual traits like leaves with different colors. With his experiments on 4 o'clock plant or Mirabilis jalapa, he found out that leaf color inheritance is dependent on maternal parent, suggesting the involvement outside the nucleus. It showed that inheritance isn't just about genes in a cell nucleus, it's also about what's inside in the tiny parts of plants called chloroplasts. This discovery of Correns led to the understanding that organelles like chloroplasts, which are responsible for photosynthesis, are inherited maternally.
Let's talk about Erich von Tschermak. 1900, Tschermak independently rediscovered Mendel's law. He was an Australian botanist. He is the best known for his independent rediscovery of Mendel's work on inheritance, along with Hugo de Vries and Carl Correns. Erich von Tschermak, along with Hugo de Vries and Carl Correns, independently rediscovered Mendel's law of inheritance in 1900. Tschermak was conducting his own research on plant hybridization and observed the pattern of inheritance that Mendel had described decades earlier. He then reviewed the scientific literature and found Mendel's original paper, realizing that His own findings confirmed Mendel's work.
Hello everyone, I am Claire Pretty Magallanes, and today let's talk about Thomas Hunt Morgan, 1868 to 1945. He was an American zoologist and geneticist, and for his contribution, he expanded the work of Mendel through discovering sex-linked inheritance and chromosome theory of heredity with the use of fruit flies. Although Gregor Mendel demonstrated the inheritance of traits, where were these qualities found? The solution was found by Thomas Hunt Morgan using fruit flies. Morgan examined eye color, a characteristic associated with sex chromosomes. An important development appeared from crossing red-eyed females with white-eyed males. The next generation of white-eyed males was far more common than white-eyed females. This wasn't explained by Mendel's work alone. Morgan's discovery was demonstrated that the eye color gene was located on the X chromosome. Morgan's discovery resulted in chromosomal theory of heredity, which believes that the genes are located in chromosomes and that inheritance patterns can be determined by how they function during cell division. By integrating physical reality existence of chromosomes with Mendel's theoretical principles, this transformed genetics. Thomas Hunt Morgan's experiments with fruit flies expanded our understanding of inheritance, which also established the framework for contemporary genetics and molecular biology.
Ronald Fisher was a British statistician and geneticist who integrated Mendel's laws with Darwin's theory. Born in East Finchley, London, he studied mathematics at Cambridge University, graduating in 1912. During his time at Cambridge, Fisher developed an interest in statistics and evolution. He aimed to resolve the debate between the biometrician, who only focused on statistical analysis for biological data, and the Mendelian, who emphasized genetic inheritance patterns. In 1818, Ronald A. Fisher published a paper titled The Correlation Between Relatives and the Supposition of Mendelian Inheritance. He reconciled Gregor Mendel's genetic laws with the statistical observation of scientists like Carl Person and Francis Galton. This integration laid the foundation for biometrical genetics, a field that combines statistics and biology to study inheritance patterns. In 1919, Fisher joined the Rothamsted Experimental Station, where he continued his research in genetics and statistics. He worked on integrating Mendelian genetics with Charles Darwin's theory of natural selection, culminating in his 1930 book, The Genetical Theory of Natural Selection, where he developed ideas on sexual selection, mimicry, and the evolution of dominance. Ronald A. Fisher made a significant contribution to Gregor Mendel's studies by critically analyzing Mendel's experiment on P+. He noticed that Mendel's results were unusually close to the expected ratios of inheritance. He suggested that the data might have been adjusted either consciously or unconsciously to better fit the theoretical prediction. This observation sparked what became known as the Mendel-Fisher controversy, a debate over the accuracy of Mendel's findings. Beyond this, Mendel's work connected Mendelian genetics with Charles Darwin's theory of natural selection, laying the foundation of modern population genetics. His analysis not only brought attention to Mendel's work, but advanced the understanding of genetics and its role in evolution.
Hi, I'm Grace Torres from AG2B. So today I'm going to talk about the contribution of Charles Darwin from Mendel's study. So this is Charles Darwin from 1809 to 1882. The primary contribution of Charles Darwin was his theory of evolution by natural selection, which he introduced in On the Origin of Species 1859. This theory emphasized how traits are advantageous for survival, reproduction are passed down over generations, and shaping the diversity of life. However, Darwin did not understand the mechanism of inheritance, which was crucial to explaining how traits were transmitted. Although Darwin's ideas about heredity, such as his pangenesis theory, were incorrect, his studies laid the foundation by studying the inheritance as a key component of evolution. Mendel's discoveries about genetic inheritance provide a missing piece to Darwin's theory, explaining how traits were passed on a predictable and stable manner through discrete units or genes. So together, Charles Darwin theory by natural selection and Mendel's properties about inheritance form the basis of the modern synthesis biology, combining genetics with evolution. And that's all.
Good day, everyone. I am Otsala Emro Jr. So now I will further discuss the contribution of James Dewey Watson. James Dewey Watson was born on April 6, 1928. He is an American molecular biologist. He is most renowned for helping to uncover the DNA structure. And his key contribution is the discovery of DNA double helix in 1953. Along with Francis Crick also a molecular biologist. So Watson's proposed the double helix structure of DNA, which explained how genetic information is stored and replicated. Their model is based on X-ray diffraction data from Rosalind Franklin and Maurice Wilkins. Reveals that DNA consists two complementary strands forming a helical structure with base pairing. So I think natako na to isa sa biochem no katong adenine, thymine, guanine, and cytosine. So adenine always pair with thymine, then guanine always pair with cytosine. So these are the four bases that make up DNA. This discovery revolutionized genetics and molecular biology, providing insights into heredity and genetic coding. So, importante kaayo ning discovery ni Watson kay aron mas dali masabtan ang structure of how amazing giunsa pag-encode ang genes inside the DNA. In relation to Mendel's work, they are related because Mendel's work helps scientists understand how genes are passed down through generations, while Watson's work helps scientists understand how genes are encoded in the DNA.